Okay, can you all hear me?
Yes.
Fine. Now I'm going to revise more than one lesson per revision session. I'm going to start with biological molecules, enzymes, and hopefully we will also include human nutrition. So that will be 3 sessions or 3 lessons in one. I'm going to start sharing my screen. Is my screen shared? Can you see it?
Yes.
Okay, very good. Now I'm trying Google Meet for the first time. I think it is efficient, but a little bit slow. Okay, so now can you see my screen? Can you all see my screen? Hello?
Yes, yes.
Okay, good. Fine. I told you that I was going to start with revising year 10, but I'm actually going to revise all year 10 and the important things in year 9, not only year 10. Okay, so give me a second. Fine. So I'm going to start with the lessons 4 and 5, biological molecules and enzymes. I'm going to finish them now. And if we have time, we will also include digestion. I'm going to start with the most important material in year 9, which is the food tests. And I'm going to start from there. So let's revise together the tests for reducing sugars. Reducing sugars here, these include glucose. It is the most popular reducing sugar that you know. Now I'm going to move quickly. This is the Benedict test, and this is the only test that needs heating. We will use, or we will start with a solution that contains the reducing sugar or the glucose, and we will add a Benedict to it. If you want exact amounts, we will add equal volumes of the reducing sugar and equal volumes of the Benedict, and we will heat it in a water bath. And this water bath should reach at least 70 to 80 degrees Celsius. Of course, you can increase the temperature, but at least 70 to 80 degrees Celsius. And the final color, if it is a positive result, if reducing sugar or glucose is present, it should be brick red. But someone remind me, what is the starting color of the Benedict?
Blue.
It's blue, okay? And the blue is going to change to green, to yellow. Finally, it will be brick red. And this brick red color is a positive result. Very good. Now let's move on and waste any time. Now we have also the test for starch. Again, I want to tell you that the Benedict test is the only test that requires heating, the only test that requires heating. And this has to be mentioned in the answer. The heating is going to be provided by a water bath because there's 1 mark for that. And this is a safety precaution using a water bath. We will not use an open flame. We will use a water bath. Good. And why is any test out of 2 marks? Because the first mark, you have to mention the name of the reagent. So you get 1 mark for writing Benedict. And then the second mark for the color change, for mentioning the starting color and the final color, from blue to brick red. And if this requires heating, this will be out of 3 marks, like the Benedict. So 0.3, you have to mention heating using a water bath. Good. Now, the remaining food tests are usually out of 2 marks only, because 1 mark for the name of the reagent and the other mark for the starting color and the final color. Now, the test for starch, this is very easy. We will use iodine, of course. And the starting color of iodine, it is orange-brown. And the final color is blue-black. And here, you don't need to use a water bath. You don't need to use heater, nothing. Good. Now, the test for protein. Again, 2 marks. 1 mark for the name of the reagent, which is the burette. The second mark for the starting and the final colors. The starting color is blue, as you can see, and the final color, if protein is present, it is purple or violet or lilac. You can write it as purple or violet or lilac. Good. Now, why do I have this picture here? Because this is a mortar and a pestle. This is a device that you use to crush solid food samples. Sometimes they will ask you, what if we have a seed? You cannot put the burette directly on the seed. I have to crush it first with a little bit of water using this mortar and pestle. I will crush it with a little bit of water. Why do I do that? To extract the soluble products. So let's write, how will you mention, how will you do a food test for everything, for protein, for fats, whatever. Number one, if it's a solid, I have to crush it. It has to be crushed because you will miss a mark for that if you did not mention it. Especially when they tell you it's a seed. Usually they use seeds as solid samples. So I'm going to get my seed. I'm going to use this mortar and pestle and I'm going to crush it with a little bit of water. Why is that? To extract the soluble products. This means that anything that will dissolve in the water will come out. And then I will filter it and I'm going to use the filtrate. Now, this is a little bit of chemistry. If this is the test tube and this is filter paper, and I'm going to filter something, what comes out here is the filtrate. And I'm going to work on the filtrate. I'm going to work on this filtrate. And then I'm going to add my burette, and the color change will be starting color blue and the final color purple. So you do not need to mention the crushing and the mortar and the pestle parts if it is a liquid. You only need to mention that you will crush the sample if they tell you clearly that it is a solid, such as a seed. I hope I'm clear. Okay, good. Now, if they tell you what is the safety hazard in the burette, anyone has a question? Okay, if they tell you what is the safety hazard, you need to wear gloves because the burette is an alkali and you don't want to come in contact with it, so you need to wear gloves. Okay, these food tests are very, very common, not only in paper 6, they can also appear in paper 4. Now, the test for fats is the ethanol test. If you have a liquid, you will not do any crushing. You will start immediately. The first thing you do is that you will number one, you will add ethanol or alcohol. It's the same thing. After you add the ethanol, you will shake it. And then after the ethanol, what do you add? Remind me. Ethanol and then what? To wake
Water.
up with me. Number one, I'm going to add alcohol or ethanol. Yes, wake up with me. Good. We're revising together. This is very important. And then we will add water. So number one, I'm going to add ethanol or alcohol, and then I will shake. And then I would add water, and then I will shake. And what will be a positive result?
Turn milky. A milky emulsion.
Very good. A milky emulsion. Okay. If you want to know what is the starting color of ethanol, ethanol is transparent. Now, again, if the sample is a liquid, we will immediately start with the food test. If the sample is a solid, especially if it is a seed, I will have to crush it using a mortar and pestle. This is the mortar and pestle. You have this picture in Year 9 notes. So first, if it is a solid, I have to crush it with a mortar and pestle, but I'm going to crush it with a little bit of ethanol because I want to extract the fat-soluble products. And then I will filter it, and then I'm going to work on the filtrate and then add my water and shake, and positive results should be cloudy emulsion, milky emulsion, whatever. Okay, so please be careful because they try to make you fall in mistakes. If you have a liquid sample, you start with the food test immediately. If you have a solid sample, just please mention that you will crush it first because you cannot immediately add the Benedict or the burette on the solid. I have to crush it a little bit with a little bit of liquid, filter it, and then work on the filtrate. I hope this is clear. Now, the test for vitamin C, I want to emphasize on this test because you all know the DC-PIP solution, but you don't know how it is done. The right way to do it is that I'm going to have the DC-PIP in my test tube, and the DC-PIP is blue in color. And I'm going to add drops of the vitamin C, one drop at a time. I will add one drop and shake, and one drop and shake. And suddenly, the color should change from blue to what? Tell me. Colors. You're too lazy to speak out. I know you know it. Okay. And we count the number of vitamin C drops. The less the number of vitamin C drops, the more concentrated. So this is exactly how it's done. I'm going to get a test tube and I'm going to start with the DCP DC pip inside my test tube. And I'm going to add drops of vitamin C, one drop at a time. And after every drop, I shake. After every drop, I shake. And suddenly it's going to turn colors. So we're done, or this is what I thought was important in year 9 in biological molecules, the food testing. Now we'll go directly to biological molecules, the supplement. Of course, I would like to revise also the biological molecules that we studied in year 9, the fats, the carbohydrates, and the proteins. Which one of the biological molecules that I just mentioned contains nitrogen?
Protein.
Protein. Okay, fats and carbs and proteins. These are the biological molecules that we studied. We also studied water, and it's important, and some vitamins and minerals. This is the whole lesson. It's a very nice and short lesson. Fats and carbs and proteins are described as being organic molecules because they contain carbon and hydrogen and oxygen. All of them contain carbon and hydrogen and oxygen. But protein has something extra. It contains nitrogen. Very good. Okay. Fats are broken down into what reminds me.
Fatty acids and glycerol.
Very good. Carbohydrates, we have several types of carbohydrates. Whatever, any one of them is broken down finally into, hmm, come on, carbohydrates, starch, glycogen. It's broken down into what? Wake up.
Glucose.
Yes, perfect. And proteins are broken down into amino acids. Now, this is year 9 material, and this is very easy. Now, what they focus on year 10 in biological molecules is the structure of the DNA. The DNA, this is important. Let me get a screen. Yes. The DNA, this is the genetic material that is found in all living cells. The DNA, it is a large molecule that is described as being a double helix. Okay? It is a large molecule made up of smaller monomers. These small monomers, in case you forgot, is called the nucleotide. So the nucleotide is the monomer or the subunit that makes up the DNA. So I have many nucleotides on top of each other. They make the DNA. And every single nucleotide is made up of one sugar and one phosphate molecule and one nitrogen base. Okay, so I have many nucleotides on top of each other, many nucleotides on top of each other. We have a fixed part and we have a variable part in all DNA. What is the fixed part? Part A or part B? What is the fixed part? The sugar and phosphate or the nitrogen base? Who remembers?
And phosphate. Sugar and
Yes,
phosphate.
the sugar and the phosphate, this is the fixed part. And this is described as the DNA backbone. I don't know where did it go. Okay. And this is described as the DNA. This part is the DNA backbone. And the variable part are the nitrogen bases. Now, we don't need to know the names of the bases, but we need to know how many bases do we have in our DNA?
4.
4 bases, yes. And they bind with each other in a precise manner. Can you remind me which base binds with which base? A, T, very good, and C bind with G. And they bind with each other by hydrogen bonds. This is all what you need to know for O level. So let me go back to the slide. Yeah. So we have the DNA structure. This is the first objective in year 10 biological molecules, describing the DNA structure. And what is it? It is a large molecule and it is made up of 2 strands of polynucleotides coiled into a double helix. And why is it coiled? So that it takes less space inside the nucleus. This is exactly how it looks like. Okay. And we need to know that it is made up of the nucleotide that I just drew a second ago. And this is my sugar, this is the phosphate, and this is the nitrogen base. And we have 4 bases, and they bind to each other specifically. A binds with C again, and base C binds with G by hydrogen bonds. And this specific base pairing is called complementary base pairing. And here is the syllabus here that states exactly what you need to know about the DNA. So if you find this box here in your year 10 notes, in case you ask me or you don't know, this is your syllabus, copy paste it. You need to know the structure. It's made of two strands that are coiled as a double helix. We said all that. Each strand contains chemicals of bases, the bonds between the bases always pair up A with T, C with G. Full names are not required. So we're done with that. And the small monomers or the subunits are called nucleotides. Okay, I said that. These are just more pictures, and these are more and more pictures. These are the bases and the complementary base pairing, and the bases are held together by hydrogen bonds. Nice and easy. Okay, nothing new. Now we will join two lessons into one, and we will remember together classification and what makes us different. Why are we all different and we're not all identical? Because we have the same sequence of bases or different sequence of bases.
Bases and different nitrogen bases.
Very good. Different nitrogen bases, and our bases are arranged in a different order. In other words, to make things clear, this is the DNA found in all our cells. Okay, this part is the same. So we all have this part the same. We all have the same sugar and the same phosphate, the same sugar and the same phosphate, the same sugar and the same phosphate. We have this backbone. This part is the same. Then what is different between me and you and her and him? The order of bases. These bases are arranged in a different order. And every three bases code for one amino acid. And we will revise this again when we study inheritance, or when we revise inheritance. That is why we are different, because the order of our bases are different. Okay, and what makes us within the same species that we have similar order of bases, but we are not exactly the same. We are different. And every three bases codes for one amino acid. Okay, I will leave it for inheritance. But it is mentioned here that the sequence of bases that holds the code for the formation of proteins, and every base triplet or every three bases code for one amino acid. So we mentioned it here, and we mentioned it in the lesson inheritance. Again, every three bases carries the message for the making of an amino acid. The DNA is not made up of amino acids. The DNA is not made up of proteins. The DNA is made up of nucleotides. This is what the DNA is made up of. The DNA is made up of this. And this is called a nucleotide. This is not a protein, and this is not an amino acid. And what is the relation between the nucleotide and the amino acid? That the nucleotide carries the message. It carries the order. It carries the how to do the amino acid. Because every three bases codes for one amino acid. And if you remember, those three bases will be copied on a messenger RNA. And then this messenger RNA will go outside to the cytoplasm, bind with a ribosome, and this messenger RNA will be translated into a sequence of amino acids. This is the lesson protein synthesis, or the part protein synthesis in inheritance. So now you are in a stage where you are combining all the lessons in one. So again, does the DNA contain amino acids? Someone
No.
give me an answer. No, no, no. The DNA does not contain amino acids. The DNA does not contain protein. But what is the relation between the DNA and amino acids?
Every three bases goes for one amino acid.
Perfect answer. The DNA carries the base triplet. It carries the code for the making of amino acids. It does not contain amino acids. I hope this is clear. Okay, let's move on. So we're done with the biological molecules year 10 because they only focus on the nucleotides and the DNA. And then you take it in more depth next year. Now, we will take also enzymes. Enzymes, again, I include the important material I thought was important in year 9 and in year 10. We will start with the definition, enzymes, because the definition says a lot of information. An enzyme is a biological catalyst that will speed up all metabolic reactions inside the body without being used up. All this is in the definition of year 9. I don't know why it doesn't go away, but it's right there. Okay, so it's a biological catalyst, means it makes reactions faster and it is not used up in the reaction. In other words, enzymes can be used again and again and again. We don't use enzymes only once. I can use them again and again and again. Enzymes are made up of proteins, Enzymes are not used up during chemical reactions. This is important. And enzymes are specific and they are found in all living organisms, because some of you are confused about where they are found. Enzymes are found in animals, and they're found in plants, and they're found in bacteria, and they're found in fungi. And these enzymes in bacteria and fungi, we use them in industries like the yeast. And next class, which is actually tomorrow, we will talk about biotechnology and the enzymes in the yeast and how they are useful. So this is the definition. This is year 9. And then enzymes characteristics. We know that they are made up of proteins, three-dimensional proteins that have a specific shape and a specific active site. And this means that only a specific substrate will bind with this active site, and a specific substrate is complementary. Enzymes, they speed up reactions, they're not effective, they can be used over and over again. This is just important. Now, the question or the start of the supplement material. What is or how do enzymes work? The mode of action. They work by the lock and key hypotheses. Here, when you describe how enzymes work, you have to mention several keywords that are very easy. We just mentioned them, that the enzymes are specific, they are made up of proteins. These proteins are three-dimensional. They have a specific active site, and only a specific substrate will bind to it. And when the enzyme binds with the substrate, they form the enzyme substrate complex. And then the enzyme will break off and it can be used again and again and again. So all these are keywords that you have to mention when you come to talk about enzymes. Just throw them to the examiner, which are, they are catalysts, they are made up of proteins, they are three-dimensional, they are specific, they have a specific active site. When they bind with a specific substrate, they form the enzyme substrate complex. Enzyme substrate complex is very important. And then they can be used again, again, and again. Active site, this is the substrate. And this method is called the lock and key hypothesis. I don't think anything is hard, and I don't think this is new to you. This is exactly what I said here. So this is the first objective in enzymes in year 10. Mode of action, how enzymes work, why are enzymes specific? So this is the answer to any of those questions. This is the answer to any of those questions. How do the enzymes work? What is the mode of action? Why are the enzymes specific? You will have to mention all that. And please don't mention that this method is known as the lock and key hypotheses. Every lock binds with a specific key, and any keyword is involved: three-dimensional, protein, complementary, specific shape, specific substrate, enzyme substrate complex. A substrate is the material which the enzyme will act on. A product is produced. The enzyme is used over and over again, etc. Any questions? I don't think anything is hard. Now, there was a question that asked about or asks about the factors affecting enzyme activity. We studied in detail how the temperature will affect enzyme activity and how pH will affect enzyme activity. There was a question in paper 4 that asked you to suggest other factors that can affect enzyme activity. So you can add surface area of the substrate, the concentration of the substrate, and the concentration of the enzyme. I would like you to add these factors. So add them somewhere in your notes. I will remind you of them later at the end of the year for the additions that we will add. So the factors affecting enzyme activity, we studied temperature and pH. I want you also to add concentration of enzyme, concentration of substrate, and surface area of substrate. Okay, here is the objective. Explain enzyme action reference to active site, enzyme substrate complex, substrate and product, the specificity of enzymes in terms of complementary shape, fit of active site with substrate. I think this is finished. Anyone has any questions? Is anything hard? Okay, I think I lost you. I'm going to share again. Are you all with me? Okay, can you all hear me? Hello? Can you hear me?
Yes, we can.
Yes.
Okay, we lost each other, but I finished talking about the structure of the enzymes. Did you all reach this? Did you all reach that? Okay. So now we will talk about the graphs. This is the important part. Are you all with me? Can you, if please, if the connection is lost, just please make me notice. Okay? Hello?
Okay.
Fine. Now, the important part, the part that always shows up in paper 4 are the graphs. Now, we have two graphs that we have to break down in detail. The graph which shows the effect of temperature on the rate of enzyme action and the effect of pH on the rate of enzyme action. Let's talk about the effect of temperature and then the effect of pH. Now, the graph, both graphs, they look like this. You have the temperature on the x-axis. That's important because this is the variable that is changing, and the rate on the y-axis. And both graphs, they have an increase and a decrease, and we break it down into 4 or 5 parts, and it is very clear in this picture. This is one part, this is another part, this is the optimum temperature, and then you have 2 more parts after the optimum temperature, and they are explained in detail in your notes. Now, at low temperature, this part of the graph here, the enzymes are not denatured. They are not denatured, but the enzymes here are slow. So at this part of the graph, the enzymes are slow. What is slow exactly? You have to mention that I have very little kinetic energy, and the enzymes and the substrates or the molecules are moving slowly, and there is a slow rate of successful collisions, little enzyme substrate complex, little product is produced, and everything is actually clear here. Enzyme and substrates have less kinetic energy, low activity, less substrate is turned into product. Now, as the temperature is increasing, all this is increasing. We are in this part of the graph, part B, and they always ask you about parts B and C. What is happening in part B? Everything is increasing. We start with the kinetic energy. I have more kinetic energy, it is increasing, faster reaction rate, more successful collisions, more enzyme substrate complex. So here I mentioned all the keywords. What do you say at C? C is very easy. Wake up with me. What do you say at C?
Temperature.
Optimum temperature. Use the word maximum. Maximum reaction rate, maximum successful collisions, maximum enzyme substrate complex. Everything is natural. And then after C, what starts to happen to the enzyme? It starts to
Denature.
denature. And again, don't please give me incomplete answers. Don't write denature and then remain silent. You need to elaborate. The moment you mention the word denature, then you have to mention that the three-dimensional shape became destroyed. The active site, its shape has changed. The substrate will not fit. The substrate is not going to be complementary. No enzyme substrate complex. No product. Okay, so this graph is very easy. Just break it down and elaborate while you are explaining it so that you take the full grade. Now, everything is written here. Okay, please, at very high temperatures, elaborate on denaturing. You have everything written down for you. When the enzymes are denatured, okay, the bonds break, hydrogen and peptide bonds break, holding the enzyme together. The active site will change. The enzymes no longer bind with the substrate. It is no longer complementary. No enzyme substrate complex, no product. The reaction will eventually stop. Okay, now this is another graph. I don't think we need to waste any more time on it. And then the effect of pH on the rate of enzyme activity. It's important to know the difference. Again, the graph looks the same. I have a part increasing here, and then I have an optimum pH, and then I have a part decreasing. In this part of the graph at A, I'm not going to say that the enzyme slows down. This is not temperature. At A, I'm going to say that the enzyme denatured because I am very far away from the optimum pH. This is the optimum pH. Any or if I am far away from the optimum pH, if let's say the pH is 8, if I am at pH 3, this is too acidic for the enzyme. So the enzyme here will denature, and I will talk about denaturing. If I am here, and the pH is 14, that is too alkaline for the enzyme, again, the enzyme will denature. So at A, I will mention that I am far away from the optimum pH and the enzyme will denature. And at C, again, I'm very far away from the optimum pH and the enzyme will denature. What if they're asking you about this increase? You will simply say, as you are moving closer to the optimum pH, the rate of the activity is increasing. But don't mention anything about kinetic energy or the speed of the molecules or any of that. You only mention that if you are far away from the optimum pH, you will denature. So you denature at the extremes of the graph, at the ends of the graph. Here, the enzyme will denature, and here, the enzyme will denature. Is it clear? Are the two graphs clear? Again, you have two graphs that look the same, but one with temperature on the x-axis, and on the y-axis, you have the rate, and one with pH on the x-axis, and on the y-axis, you also have the rate. And both graphs look the same. You have graphs that look exactly the same, but what you see is very different. This is the optimum temperature. Here, this temperature is low, it's cold, so the enzymes slow down. Very far away from the optimum temperature, the enzymes denature. This is when you are talking about temperature. When you're talking about pH, when you're far away from the optimum pH, enzymes denature here, and they also denature here. So are both graphs clear? In temperature, you mentioned kinetic energy, and you mentioned rate of successful collisions. You mentioned enzyme substrate complex. This either increases or decreases. In pH, you don't mention kinetic energy at all. No kinetic energy, because kinetic energy here is the energy of motion that is only affected by temperature. Are the two graphs clear? Any questions? Okay. Did I lose you or are you with me? Hello?
Yes, we're with you.
Okay, so we're done with biological molecules, and actually we're done with biological molecules and enzymes. But year 9 has very important practical experiments. So this is from year 9 experiments. One of the most common experiments is the enzyme catalase that breaks down hydrogen peroxide into water and oxygen. Hydrogen peroxide is a waste product of metabolism that has to be removed. Catalase is an enzyme that is found in all living organisms. In our investigation, the source of the catalase will either be a plant or a liver, a piece of liver. And the plant that they always use is a potato. They always use a potato. Now, usually the experiment looks like this. We will put the potato and hydrogen peroxide, and we will allow the catalase in the potato to break down hydrogen peroxide into water and oxygen. And then we will see bubbles. These bubbles are due to the release of oxygen. This is the oxygen. And we count the number of bubbles. Of course, is this accurate or not accurate?
Not accurate.
It's not accurate. Now, to improve that, either we use a gas syringe or an inverted measuring cylinder like this one. Now, the other experiment that is very important, which is studying the effect of amylase and its effect on the breakdown of starch. Now, remind me, amylase breaks down starch into what? Starch is broken down by the amylase enzyme to give me what?
Maltose. Maltose.
Or any reducing sugar. You're very good. You're very correct. Now, this experiment is very common and you never memorize it. I don't know why. Now, this experiment involves 3 steps, and the picture is right in front of you. Step number 1 is that I will get 2 test tubes. One test tube will contain the amylase enzyme, and the other test tube will contain the starch. And I'm going to put them in a water bath, as you can see, for 10 minutes. Why do I do that before I start mixing them together?
To reach equilibrium.
Reach equilibrium.
Exactly, to reach equilibrium. So step number one, I'm going to get the two separate test tubes, one containing the enzyme and one containing the starch. I'll put it in a water bath and I'll leave it for 10 minutes to take the temperature of the surrounding water or to reach equilibrium, just like you just said. And then step number two, I will mix the starch and the enzyme and I'll put it back. Why do I have to wait to reach equilibrium? And if I mix the enzyme and the starch, they will react. I don't want them to react. I want them to first reach the temperature that I want, and then I mix them. Now I will mix them, put them back in the test tube. And then step number three, I'm going to get a spotting tile. This spotting tile is a tray that contains holes, as you can see here in the picture. And what do I have in this spotting tile? I have iodine. The original color of iodine is yellow-brown, as you can see. I'm going to take one drop from this mixture and put it on my spotting tile every 10 seconds. Now, if the color changes to blue-black, this means that the starch is broken down or not yet.
Not broken down.
Not broken down. I will keep on repeating this. I will take a drop out of the mixture every 10 seconds until the color remains what?
Orange brown.
Very good. This means that all the starch has been broken down. Now, this experiment is very common. It is highly repetitive in this lesson, and you never need to memorize it. I don't understand why. Again, we are studying the effect of the amylase enzyme on starch, and we're studying the effect of temperature on this enzyme. We know that the amylase will break down starch into a reducing sugar, maltose or glucose or whatever. Step number one, we will get a water bath and we will get two test tubes, one containing the amylase enzyme and one containing the starch. We will use equal volumes of enzyme and starch, and we'll put it in a thermostatically controlled water bath. I will leave it for 10 minutes to reach equilibrium. This is step number one. Step number two, I will mix these two together and I will put them back in my water bath. So now my test tube contains the amylase enzyme plus the starch. And then this is step number two. Step number three, I'm going to get a spotting tile containing iodine. This is iodine here. The iodine is yellow-brown. I'm going to get one drop out of my mixture and put it on my spotting tile every 10 seconds. And the color, if it changes to blue-black, and starch still did not get digested. If the color remains yellow-brown, then this means that all the starch has digested and this is the end of the experiment. So I'm going to measure the time needed for digestion. The shorter the time, the faster the reaction. Is this clear? Anyone have any questions over this experiment?
No.
Okay. Oh, no questions. Good. Now, the other experiment that is very important and very, very similar to what we said before, the effect of pH on the amylase enzyme. pH. So we will not get a thermostatically controlled water bath. We will change the pH. How do we change the pH? What do we use? Different what?
Buffer solutions.
Buffer solution.
Exactly. I'm going to also get two test tubes, one containing the starch alone, one containing the enzyme and the buffer, and then I'm going to mix. And the moment I mix is the moment I'm going to time. I'm going to use a stopwatch, and every 10 seconds, here is my timer, and every 10 seconds, I'm going to get one drop out of my mixture and put it on my spotting tile. If it is blue-black, then starch is still not digested. I will wait for another 10 seconds, and then I will take another drop, and another drop, and another drop. And then the color here is starting to remain orange-brown. When do I stop? When the color of the iodine doesn't change. When the color of the iodine remains yellow-brown, this means that all starch has been digested. So I'm going to record the time needed for the iodine to remain yellow-brown. The shorter the time, the faster the reaction. Is this clear? Okay. Now, in the first experiment, what can be my control? In both experiments, actually, what can be my control? Anytime I use
Boiled enzyme.
very good. Boil the enzyme. In this experiment, where I will use a potato as my source of enzyme, what can be my control? Boil the
Potato.
potato. Yes. So if you want a control and you have any living tissue in the experiment, whether a plant or a potato or whatever, I will boil it. Good. Also, in the temperature of the pH, I can boil the enzyme, or I can have a pH of what? Neutral pH. This also can be a control. Now, sometimes they ask you, why don't I add my iodine directly on the mixture? Why do I have to take a drop out and put it on a spotting tile?
Will destroy the solution.
Yes, I'm going to destroy the mixture. If I add an iodine on the mixture, then I cannot use it again. Good. So here we're done with 2 lessons actually. Here we talked about the amylase and the protease. I'm going to revise this again when we do the digestion. I'm done. But I'm not done. No, I'm done with enzymes and biological molecules. I'm going to also revise digestion, because digestion also is very short. Any questions? So in one revision session, we will revise minimum 2 lessons. I'm going to finish digestion also very quickly, because you will realize how easy it is. Now, again, I included the importance year 9 material and year 10. In digestion or human nutrition, they start with the importance of bile. The first objective in your syllabus is to outline the role of the emulsification or emulsifying of fats and oils to increase the surface area for chemical digestion. This is the first objective. Now, the emulsification of fats, who is responsible and what is the emulsification of fats? Who emulsifies fats?
Bile.
Remind me. The bile. Bile is a waste product. Now we are studying excretion, and we know that bile is a waste product, but it's a useful waste product. I'm going to use it before I excrete it. Let's study the history of the bile. Bile is the result of the breakdown of all red blood cells. I'm going to break down all red blood cells in the wrist and the liver and excrete bile. So it is produced in the liver and stored in the gallbladder. This is my liver and this is my gallbladder. Now, bile is used in digestion because it helps in the physical breakdown of fats or the emulsification of fats. Emulsification of fats. Emulsification is the physical breakdown of fats. In other words, fats will be broken down. Fats will be broken down into smaller pieces or smaller globules. And why do we physically break down fats? Tell me.
To increase the surface area.
Very good. To increase the surface area for who? Who am I helping? Who will work next?
Enzymes.
The enzymes. Because fats are broken down over 2 stages. Number 1, physical breakdown using bile. Bile will break down the fat droplets, the big droplet, into smaller droplets. And this will increase the surface area. And then who will work next? Like you said, the lipase enzyme that will break down the fat into fatty acids and glycerol. That's it. So going back to the history, bile is the result of the breakdown of old red blood cells in the liver. And then bile is going to be stored in the gallbladder. Bile has 2 functions. One function, the physical breakdown of fats, emulsification. It's called emulsification. Fat is a big droplet. I'm going to physically break it down into smaller droplets. This will increase the surface area. And then this is going to help the enzyme lipase to continue the digestion of fat and chemically break it down into fatty acid and glycerol. I'm done. This is emulsification. The second function of bile is that it is an alkaline mixture and it contains sodium bicarbonate. And what does it do? It helps in the neutralization of the acids coming from the stomach. Remember the stomach? And remember that the stomach produces hydrochloric acid? Remember? Okay. Where is we have here, yes, this is my stomach. The stomach here digests protein. In case you forgot, let's do a quick revision. Salivary glands, saliva, they contain an enzyme that breaks down starch. What's the name of this enzyme? Remind me, because I forgot. Good girl. Amylase. The amylase will break down starch into what?
Maltose.
Good girl. Not glucose. Maltose. Excellent. And then the food will move down the esophagus. How does it move down the esophagus?
Peristalsis.
Yes, excellent. Peristalsis, peristalsis or peristalsis. These are series of contractions. And then the food will reach the stomach. What is the main nutrient digested in the stomach?
Hydrochloric acid and pepsin.
Okay, this is what is secreted by the stomach. Yes, the stomach digests fats or proteins or carbs.
Proteins.
Yes, you're correct. Protein. And proteins are broken down by the pepsin, like you just said, and the HCl is produced by the stomach to provide the acidic media for the pepsin and to also kill any bacteria or kill any pathogens in your food, kill the bacteria in your food. So I'm explaining all this to remind you that the food coming from the stomach and entering the first part of the small intestine, which is the duodenum, this food is acidic. We have enzymes in the small intestine. These enzymes, they don't like acids. They like an alkaline medium. So this is one of the functions of the liver. The liver has two functions. One, it produces bile. And bile helps in the physical breakdown of fat or in the emulsification of fat. Liver has another function. What is the second function? What does it do to the acids coming from the stomach?
Neutralize it.
Very good. So this is the second function of the liver, neutralization of the acids coming from the liver. Why? Because it contains sodium bicarbonate. And this is the neutralized or the alkaline agent that will neutralize. And the emulsification of fat is achieved by the bile salts that are found in the bile. Okay, I hope I reminded you with this part. And then we will reach the duodenum. The duodenum, this is the duodenum, the first part of the small intestine. The duodenum, any digestion happens in the duodenum, or is it only a receiver?
Only a receiver.
Very good. The duodenum receives two things. It receives one, bile, and we discussed it in detail. What else does the duodenum receive?
Pancreatic juice.
Perfect. It receives juices from the pancreas called the pancreatic juices. And these juices contain a collection of enzymes, all the enzymes that we studied. The amylase, the pancreatic amylase, the maltase, trypsin, not pepsin, pepsin in the stomach. Trypsin is produced by pancreatic juices and also produced in the small intestine. And lipase. And I think you are very familiar with those enzymes. What does the maltase break down?
Maltose.
Maltose to glucose. What does the trypsin break down?
Proteins.
To amino acids. And what does the lipase break down?
Fats to fatty acids and
Into
glycerol.
fatty acid and glycerol. So it is actually a very interesting journey. You will first eat through your mouth. Your teeth will help in physical digestion. Your saliva will help in chemical digestion. Your salivary glands or saliva contains the amylase enzyme that will break down starch into maltose. The food will move down the esophagus by peristalsis. It will go to your stomach. The stomach will also do physical and chemical digestion. It will do physical digestion by the churning action of the stomach. The stomach is a muscle. It contracts and relaxes and contracts and relaxes. Also peristalsis. This is called churning action. Okay, and also the stomach produces pepsin to break down proteins and hydrochloric acid to kill the bacteria and provide the pepsin with the optimum pH. And then after the food is finished in the stomach, it will enter the duodenum. The duodenum is the receiver. It will receive bile from the liver that will help in the emulsification of fats, and it will receive pancreatic juices from the pancreas that contain all the collection of enzymes. All this is going to go to the small intestine. The small intestine will also have two functions. Function number one, it will finish off digestion, the 10% of the digestion remaining, finish off digestion. Again, the small intestine, the epithelial lining of the small intestine will also release all the enzymes that we mentioned, the amylase and the maltase and the proteases and the lipases, everything will be released, everything will be digested. After the food is 100% digested, what does the small intestine do to this digested food?
What?
Very good. It will absorb it. and we will talk about the absorption part, and we will be finished. Now, what is the function of the large intestine? Can someone remind me?
Absorption of water.
Very good. It absorbs water and it stores the feces in the rectum until it comes out of the anus. And when the feces comes out of the anus, it is egestion, which is a form of excretion. Wow, we are revising a lot of things. Good. Okay. Now a question. Which will absorb more water? The small intestine or the large intestine?
The small intestine.
Perfect. The small intestine. You're amazing. Okay. So we actually revised all this. I'm very, very happy. Good. So fats, we said they are broken down, emulsification, but the function of bile, we said it, physical breakdown of fat, increases surface area to help for the action, to help the lipase enzyme does its action. And the bile, the second function is neutralizing acids coming from the stomach. And we finished this objective. Very good. Then we talked about all these enzymes, the saliva, the pancreatic amylase, maltase, pepsin, and trypsin. Tell me, what is the optimum pH in the saliva?
7.
Yes, in your stomach.
Acidic.
Yes, I know that you know. And in your small intestine,
Alkaline.
slightly alkaline. Good. So we're done with these two objectives also. And we did all that. And we, of course, know that all the digested food will be carried by the hepatic portal vein to go to the liver. And the liver will do some filtration and assimilation. And then it will be distributed to the whole body. Very good. Now we just finished the duodenum, discussed it while we were speaking. Don't forget that the gallbladder stores the bladder and the stores the bile. And the bile is carried by the bile duct to go to the duodenum. This is the duodenum. This is a very nice picture of your duodenum. It's actually in your notes. And the pancreas also releases pancreatic juices carried by the pancreatic duct. This is the pancreatic duct. Okay. The bile contains any enzymes?
No.
No.
No. Only bile salts and sodium bicarbonate. Okay. All this is done. Now, the last thing that I'm going to revise with you, and then I'll let you go. We revised all those enzymes, very good, is how the small intestine or the ileum is adapted for absorption. This is also a clear, I don't know what happens to my PowerPoints, they run away. Okay, they're here. So see, this is the last objective, objective 3, 4, and 5, and I'll leave you. Explain the significance of villi and microvilli in increasing the internal surface area of the small intestine. Objective 3, objective 4, describe the structure of the villi. Objective 5, describe the roles of the capillaries and lacteals in the villi. So this is the small intestine. We said after the food is 100% digested, it is absorbed. Why is the small intestine very good in absorption? Remind me. What does it have? What are these foldings? Now this is so easy.
Villi.
Come on. You want villi. And these villi, they're there to increase the what?
Surface area.
Surface area. And this is one big folding, it's the villi. And the villi are lined by what?
Microvilli.
Yes, you are correct.
Epithelial cells.
Epithelial cells, correct. Excellent. That contain microvilli. So don't get confused. This is one villi. One folding is one villi. And the microvilli are the smaller hairs that are found on the epithelial cells. Very good. Now, what is found inside here?
Capillaries.
Yes, you have capillaries, very good, to absorb the digested food. And you have a central vessel here that absorbs the fatty acid and the glycerol. What are they called?
Lymphatic.
Lymphatic.
Very good. The lacteals or the lymphatics. Perfect. We actually nearly finished. Also, other things that help in the absorption process. The walls are very, very thin or thick or wet.
And
Thin.
then for shorter
Very thin. They are one centimeter for faster absorption. Excellent. Also, they do things or they absorb materials by diffusion and active transport. So for
So
active transport, what does
mitochondria.
the wall contain? Lots of, what did you say, my dear?
Mitochondria.
Perfect.
have a lot of
Mitochondria and the cell membrane
Carrier
proteins,
proteins.
everything. Carrier proteins. Lovely, lovely, lovely. This is a picture here from an exam. A is the what? What's A?
epithelial cells.
Good girl. B. Lactose.
Lactose.
Lactose that absorb the fatty acid and glycerol and the fat-soluble vitamins. Let's see.
Capillaries.
The capillaries. Amazing. So we just did and we finished objective 3, 4, 5. And it's all written here. Okay. And we said it. Good. We have also dense blood capillaries to maintain a concentration gradient and to absorb, and it's long to increase surface area. Everything is written here, and you said everything. Now we're done with year 10. I thought that the deficiency diseases in year 9, they were important also. Quick revision. Survey, it's because you're not eating enough what? And you look like this.
Vitamin C.
Vitamin C.
Vitamin C, you have bleeding and inflamed gums, and your teeth are not very clean. Vitamin C is needed for healthy gums, healthy tissues, proper healing, important for immunity also. Now, this is what we don't have or we don't eat enough.
Citrus fruit.
No,
Vitamin
this,
D.
this.
Vitamin D.
Vitamin
And
D.
calcium.
Yes, vitamin D and calcium, they're friends, they work together. This gives you rickets in children and weak bones or osteomalacia in adults. You have bone pain and your bones don't grow, and soft and weak bones. Okay, vitamin D is very important for the absorption of calcium. That's why they both have to be together. Okay, vitamin D is stored under the skin, in the fat under the skin. It's a fat soluble vitamin and activated by the sun. And vitamin D is important for the absorption of calcium. Okay, good. Now, vitamin A is important for immunity. What's important is iron. Iron is needed for the making of what?
Hemoglobin.
Hemoglobin.
Hemoglobin. If you don't eat enough iron and you don't have enough hemoglobin, you will suffer from
Anemia.
Anemia.
Anemia means less hemoglobin, less oxygen, less aerobic respiration, less energy. Signs of anemia, they usually ask you. You're always tired because you don't have enough energy. You don't focus, you have headache, your skin is very pale, you're yellow, loss of hair, brittle nails. Why do females suffer from anemia more frequently than males? Because they lose blood.
Menstruation blood.
Yes, menstruation. Okay. I'm done with the question. So I hope the revision was effective. We revised experiments we revised the food tests, we revised biological molecules and DNA, we revised the experiments that involved the breakdown of starch amylase. This was important. We revised digestion, bile, and we revised deficiency diseases. So we revised actually 3 lessons, 9 and 10. Any questions?
Excuse me.
Yes, my dear.
I had a question in paper for excretion.
Okay, send it to me privately
Okay.
now. Okay, my dear?
Thank you. Yes.
You're welcome. Okay, see you tomorrow. Tomorrow we'll start a new lesson. We will start genetic engineering. And get paper 4. I'll tell you what to get on WhatsApp. Bye.